Hey guys, welcome back to another video for our Stage 6 Biology course where we're looking today at Module 2, Organization of Living Things. Now, if for whatever reason you come across a fact or a figure that you're not sure of, please ensure that you contact your biology teacher or leave something in the comment section below. So, for this video we're going to start off with a very long topic, looking at unicellular and multicellular structures. So already we're comparing prokaryotic cells and eukaryotic cells. If you watched video 1 on module 1 for this course, then we already know that prokaryotic cells have no membrane-bound internalized structure. So essentially, imagine a house. Most houses are like eukaryotic cells. Each room hosts a specific series of tools or built-in structures for a certain purpose. A prokaryotic cell is more like everything is all in one big room and there's no dividing thing between them. Now, if you've ever gone to a house like this before, or you've been out to someone's shed and they had a toilet in one corner of the room and another corner of the room they had a kitchen sink, you know it's not a particularly organized place, and in essence it becomes very, very disruptive. Now, the reason that eukaryotic cells have these internalized barriers that prokaryotic cells don't is to increase efficiency. If every cell is required to produce a certain amount of energy or get rid of a certain amount of waste product or utilize a certain amount of goods and products, then naturally they have to be able to keep pace. And in order to keep pace, they have to be organized. In order to be organized, you need internalized structure. So it's much like how in our schools we have organized structure in order to make things more efficient and cause less problems overall. Now, obviously, if you'd like at any point, you can pause the video and copy down the notes that are included here. Otherwise, you can look them up on your own. Fact check me. See if anything I've said doesn't make sense or I might have said something incorrect. Then let me know. I'm always happy to be corrected. So here we compare unicellular organisms and multicellular organisms. Now, typically, people often get prokaryotic cells and eukaryotic cells mixed up with being just unicellular and multicellular. It's a very heavy correlation, but there are a few exceptions to the rule. Okay, the unicellular organisms have a very high surface area to volume ratio, which means they can exchange materials very easily. They don't need to coordinate with other cells around them, they can just do it whenever they want. So, 99 times out of 100, they're going to be prokaryotic cells. Multicellular organisms that have multiple cells that need to communicate to each other and work together in order to benefit the organism as a whole will utilize eukaryotic intercellular organization. All right. Now, we also talk about how surface area to volume ratio is organized. Imagine here we have a whole bunch of blocks. Now, if we're using a block that is 1 centimeter by 1 centimeter by 1 centimeter, it has a total surface area of 6 centimeters squared. Okay? Now, the total volume is 1 by 1 by 1, obviously. So we have a surface area to volume ratio of 6 to 1. That's a pretty high ratio. But imagine we scaled it up. Let's say instead of being 1 by 1 by 1, it's 5 by 5 by 5. Now, in doing so, the total surface area here is 5 times 5 is 25, times that by 6, that's approximately 150 centimeters squared. That's massive. But compare that to the internal volume. There's a lot of mass inside there that's not exposed to any outside surface. That's 125. So instead of being a 6 to 1 ratio, it's actually a 1.2 to 1. That's dropped a great amount. But look at what happens if we were to cut this up into our 1 centimeter boxes. If we were to cut this up, we've exposed tons of the internalized structure inside of it to the open air. Now, it's for this same reason that when you're starting a campfire, you don't start by just tossing one giant log on there and lighting it with a match. That's not how it works. Instead, you start with large amounts of very small products like kindling or twigs or leaves, and then you put on progressively larger and larger pieces of material, because the energy has to be able to overcome the surface area and get into the internal part of the fuel source. Now, when we cut this up, the total surface area has changed into 750 centimeters squared. All right, all of these boxes inside have also been exposed. The total volume of each box, or total volume of all the boxes, is still the same, but the ratio has shifted back to match our initial one here. So, what does this all mean? What this means is that even if you have a massive organism like an elephant or a grizzly bear or a whale, their cells are not larger than ours, just as our cells are not larger than a mosquito or a fly's. The reason why is because the surface area to volume ratio has to be the same in order to ensure that materials can get in and out of a cell at a reasonable rate. So what this means instead is that larger organisms don't have bigger cells, they just have more of them, and they have more infrastructure in order to ensure that every cell can take in nutrients and remove waste and get dissolved gases where they need to go. Okay? Now, we know for a fact that not every cell in your body is exactly the same. When a sperm and an ovum meet for the first time and form a zygote, the next few months are simply cells we call stem cells. Now, these cells over time will naturally progress and diverge into specialized types. For instance, we have muscle cells, we have fat cells, bone cells, blood cells, nerve cells, epithelial, immune, and sex cells. And each of these cells looks very different to the other ones because they're all designed for a specific purpose that helps the organism as a whole. Think of it like comparing a soccer team. Now imagine that this team has been playing from when they were in kindergarten to when they get into the Premier League. When they first start out in kindergarten, they're the stem cells. Every one of them does the exact same thing, which is they all huddle around the ball and they follow it around like a swarm of flies. It's hilarious to watch. It's the best thing in the world. But as they get older, they start to realize that they have different jobs. So then instead of just having a whole bunch of kids just following a ball, you have defenders, you have attackers, you have wingers, you have a goalkeeper, you have a striker, you have midfielders, you have reserves. And then all of a sudden they become rigidly defined by those roles. And so when someone asks them, oh, what do you play? And they say, I'm a midfielder. They don't say they're just a soccer player. They give a very specific role of what they do on that team and how they contribute to the team as a whole. So each one of these designs is based upon how it's meant to work. Look at the nerve cell down here. It looks almost like a tree almost with tons of branching dendrites. The reason why is because part of its role is communication. It needs to connect with hundreds of other nerve cells around it. So by having more branching off, it can connect to more nerve cells, which means it can do its job better. Blood cells are biconcave in nature and lack a nucleus. Reason why is because they're trying to increase the surface area so the hemoglobin inside of it can connect to more dissolved oxygen. Every one of these is designed in a way to help it do its job better. Okay, look like the goalkeeper. The goalkeeper is not going to have the same aerobic capabilities as, say, a midfielder. Midfielder is running around left, right, and center, trying to ensure that everyone's, focused on the game ahead. The goalkeeper just has to be able to dive left, dive right, or move around inside the box. He doesn't have to run from one end of the field to the other one, not as much as the midfielder does. The wingers might be more developed in their ability to control the ball on the left or the right hand side as opposed to, say, the defenders, which have to be able to do both of them more regularly. So, if we look here, we have a longitudinal section of a shoot apex showing the location of meristems and local and young leaves. So, when a plant cell starts off, it has very undefined cells to start with. But over time, as they become more and more developed, they become more specialized. Now, here we can compare different types of tissue. Now, we know that cells, when they connect together, they make tissues, and those tissues make up organs. Those organs make up systems, and those systems make up a multicellular organism. Okay? So here we have four main types of animal tissue: epithelial, connective, nervous, and muscle. Each type of these types they come in various shapes and sizes. Epithelial is essentially a membrane that prevents things from crossing over it or controls entry and exit. So like your skin, every cell inside the body has a cell membrane around it. But when put together, those tissues can work together to contain fluids, or they can prevent something from entering where it shouldn't. Connective tissues hold you together. Nervous tissues send messages, and muscle tissue either extends and relaxes or it contracts, allowing motion. The four main types of plant tissue are meristematic, vascular, dermal, and ground tissues. So, vascular tissues is designed to transport materials. Dermal tissue is like a skin. Ground tissue is designed to help anchor it. And meristematic is designed to branch out and increase surface area, often like leaves. The arrangement of cells into tissue, organs, and systems provide multicellular organisms with abilities that are beyond a single-celled organism's capability. Single-celled organism looks at a job and says, oh wow, I couldn't possibly do that on my own. Multicellular organism goes, don't worry, I've got my mates with me. We can work together to achieve this goal that the single-celled organism couldn't. Organic organization. Now, we already talked about this. This is the kind of thing you would have done in year 7. We start with the organelles, such as mitochondria, nucleus, vacuoles, all that stuff. You go up to cells, tissues, organs, organ systems, and the organism as a whole. Now, by going any smaller than an organelle, technically you're going to the point of which it's no longer alive. Okay? If you're ever unsure if something is alive or dead or not, use the Mrs. Gren mnemonic. If you're unsure about that, ask a biology teacher or ask a year 7 science teacher, because they're the ones that told you. Now, autotrophic structures. When we're talking about autotrophs, 9 times out of 10, we're talking about plants. Now, in order to understand exactly what's happening inside of an autotroph, we need to have the right scanning technology. And this can vary in different styles. So let's have a look at what type of technologies are available. New technologies can be used to produce 3D images of structures and pathways involved in the movement of products of photosynthesis. Radioisotopes are one possible example. Radioisotopes are a form of element that emit radiation, which can act as tracers and follow pathways used in making photosynthesis, detected via a number of means. They were used to determine that O2 produced in photosynthesis came from H2O rather than CO2. Carbon-14 added to CO2 supply traced the movement of glucose produced through the oxygen, through the plant. Now, in the last video we looked at respiration and photosynthesis. Alright, now a radioisotope is a type of atom that is relatively unstable because it has an excess number of neutrons. Think of it like an unbalanced form of a Jenga tower. Now naturally, this Jenga tower wants to be stabilized, so it tends to leave off parts of itself in order to remain balanced. A radioisotope works in the same way when it releases neutrons into the environment. Now this we often detect as a form of radiation. Every organism has a certain amount of radioactive material inside of them. Humans, plants, animals, fungi, we all do. In fact, in order to date mummies, we often, or other organisms, we tend to use a carbon-13 isotope, which is one extra neutron that it should normally have. The problem is, though, that this technique only works with stuff that's significantly old. In other words, more than 5,000 years old. Anything younger than that, and it can't accurately date it. Now, when we talk about radioactive isotopes, we're also talking about a half-life. Just a quick example. Get a page in front of you. Now, imagine I have this A4 sheet that represents 100% of the radioactive material when that organism was still alive. Now, over a set period of time, that radioactive material is going to be halved. So let's say it's 500 years. 500 years in order for 50% of that energy to have been dissipated into the surrounding environment. That's up to 500 years. Now, after another 500 years, it's been halved again. So we started at 100%, we went to 50, Now we're at 25. Now another 500 years have passed, and it's been halved again. .5%. Now another 500 years have passed. 6.25%. Now if you were to graph this, it would look like something similar to a parabolic curve or an exponential curve on its side. The reason why is because by halving and halving again over a set period of time, you've induced a half-life. Now different elements will have different lengths of half-lives. For instance, uranium might be several million years. Other radioactive isotopes might be a few hours. Some are only a few minutes long. But each one is different, but they work the same way. After a set period of time, the radioactive potential has been halved. Same length of time again, it's been halved again, and so on and so forth. Now, when we talk about plants and their structure, we often refer to what's known as the root and shoot systems. Now, while it sounds like a very horny cowboy, it's in fact two distinct layers. The root system is any part of the plant that is extended underneath the ground. The shoot system is anywhere where it has breached the surface of the ground. So looking at this diagram, we can tell what parts of the root and shoot systems. Now, for the most part, the plant portion that is above the ground as part of the shoot system is responsible for obtaining sunlight in order to convert carbon dioxide into glucose. Now, in doing so, it has to transport what it has created down the plant. Now, the root system, aside from providing structure and anchorage for that plant, also absorbs water and other nutrients for the plant to grow, and it transports it up the plant. But how do you reckon they move them up and down? They move them through these xylem and phloem tissue. Think of them as like arteries and veins inside of a plant. A xylem tissue transports water and dissolved material up the root system, and does this through like a capillary style action. So if you place a paper straw into water, over time the water will move up the straw because it's being soaked up by the dry straw material. Pardon me. Now, in doing so, it reaches the rest of these plants. However, the phloem system sends sucrose and sugars down the plant using gravity. For this reason, many plants are able to orientate themselves so the plants are always growing up. Even if they're growing out of the side wall, they will orientate themselves so they go up. Very unique thing to look at. Now, it would be advantageous to copy down this image, particularly with all the terms on it. Please pause the video and ensure you get it down. Now, the root and shoot systems both require a large surface area to volume ratio to absorb materials from the surrounding environment. The shoot system uses leaves that are extended over a wide, flat area in order to absorb sunlight and exchange gases. The root system has many lateral roots and root hairs coming off it to increase surface area to absorb water. So already we're coming across some very unique terms here that will be related to what we see in this image. Please ensure you copy them down. Now, gas exchange is important for every living thing. It doesn't matter if you're an animal, vegetable, or fungi. Now, in plants, gas exchange occurs through the stoma and lenticles. Now, imagine that you've got a leaf. If you're looking at the top half of a leaf, particularly Australian plants, they have a waxy coating and they're darker colored. Flip it over and you can see tiny pores that are not necessarily the stomata, but they do aid in releasing gases, and it's much paler. The reason why is because Australia is so hot and dry, we want to ensure that we don't lose too much of the gas that we're producing, including water vapor. So in doing so, it's almost like the plant has evolved to have sun protection. Now, Guard cells control stomata opening, fill with water to bend open and close, lose water to straighten and close. As such, plants need to balance their need for gas exchange and water conservation carefully, opening the stomata in the dark and blossoming in the light. So, like many Australian organisms, plants are going to release gases when it's cool enough that they can control the release more carefully. When it's daytime and it's suddenly much too hot, they close them up and wait again until it's darker and cooler in order to operate properly. Now, in animals, we have alveoli. Imagine a bunch of grapes. Now, the stem are part of what we call the bronchioles. Now, these are branching and branching and branching portions within the lungs, and they're each tipped with alveoli, tiny bubbles that have a very, very thin layer. All the blood vessels flow around them and exchange carbon dioxide and oxygen across them. When we breathe in, you're taking in oxygen to store inside the lungs. That oxygen is being dissolved into the bloodstream. But at the same time, the bloodstream is kicking off CO2 and slowly filling the lungs, meaning you have to breathe it out. And so it's a constant process of breathing in, breathing out that you've been doing the whole life, and you've been absolutely killing at it. So great job for breathing. Fish are a little bit different to most terrestrial animals in the fact that they have specialized structures called gills that absorb small amounts of dissolved oxygen in water. And they're clipped with blood capillaries. So they don't have lungs, they have gills. Some fish, like sharks, have what we call ramjet gills, which means that they have to swim forwards in order to breathe. If a shark stays still or is in still water, it could suffocate, which is horrible, but really interesting. It also means that while you buy an aquarium, you also have to buy a water filter that's able to pump in bubbles of oxygen dissolved inside the water. Insects are a little bit different. Insects exchange gases via pores called spiracles, which lead to tracheal tubes, which then branch into smaller tubes called tracheoles. The tracheoles bring air directly to and from cells of the insect. Therefore, blood is not involved in transporting gases. This only works because insects are so small, they can transport gases really easily. So instead of them having to dissolve it inside blood and then transport to every cell in the body, they just do directly from the air. Now, in the structure and function of plants, here we have the composition of xylem and phloem. Ensure that you pause the video and copy down that information about both of them. The scientific understanding of plant structure and function has developed by scientists over many, many years. Hypotheses, theories, and models are changed if contradicted by new evidence. Many scientists have contributed to the understanding of photosynthesis and transpiration cohesion tension theory. It's like everything in science. If a new idea has evidence and contradicts an old idea, then the old idea needs to have overwhelming scientific evidence that has been peer-reviewed in order to contest the new one. If not, and the new idea makes more sense, answers more questions, fits more neatly with the evidence that's applied, old idea removed, new idea installed. Alright, so let's have a look at some of the xylem and phloem material. Xylem is comprised of vessels and tracheids, which are long, thin, continuous tubes made of dead, lignin-reinforced walls. Material is transported up the plant via transpiration cohesion tension theory. As water evaporates out of the leaves, it draws up water from the roots via water cohesion and wall adhesion. So like our paper straw in the glass idea. It's dry at the top, it's wet down the bottom, the water will move up in order to wet more of the straw. And because water holds onto itself quite well, it drags up other water with it, so there will be a decrease in the water level over time. A small amount of root pressure forces water already present in the xylem vessel upwards. So it's like how you could siphon water out of a hose by standing up above it. The hose is, lined with dry paper inside of it. Breathe in. The water will naturally want to go up because it wants to wet the paper, but it's also been sucked up due to suction. Phloem is composed of sieve tubes, lined tubes with sieve plates at each end, moving glucose via translocation, and companion cells, which assist the sieve cells. Glucose is moved by source sink theory, as the sugars move from high pressure region to a low pressure region. So in essence, huge amount of glucose here, not much glucose here, naturally starts to move until both of them are equal. But as the plant grows, it will need more, use more of the fuel, so this one goes back down. This one builds up as it takes more material in and produces more glucose. And so they try and equalize again. And it continues this dance for a long period of time. Alright, digestion in animals. Now, here we have a comparison of mechanical and chemical digestion. If you're not great on understanding stuff, please ensure you have that table down. Now, digestion is used to break down food into particles small enough that cells can use. And often this means it has to be dissolved into the bloodstream. Now, this consists of two processes. One is mechanical digestion, whereby you physically break food down. For instance, chewing. Chickens, for instance, don't have teeth. So occasionally a chicken will eat a stone and it will help mash up food inside of its stomach for the same effect as us using our teeth. Now, in breaking it down to smaller pieces, we increase the surface area to volume ratio. This enables the next step, chemical digestion, to take place. So in chemical digestion, we're adding stuff like acids and enzymes in order to help break down the food into even smaller pieces. The smaller it is, the faster it can be absorbed, the quicker it can be used to help cells. So for here, proteins are broken down to amino acids, carbohydrates are broken down to simple sugars such as glucose, and lipids are broken down to glycerol and fatty acids. Here we have a comparison. Now let's say we're looking at a deer and we're looking at a fox. A fox is a carnivore. It eats protein primarily from stripped animal flesh. A deer is a herbivore. It eats plant life such as grasses. Now because grasses are more complex and contain long-chain carbohydrates, it needs more time to break down. So it uses a 4-chambered stomach with a large rumen, so it can have various stages of breaking stuff down, and then a very long intestine, both long and short, cecum, and a spiral loop here, in order to absorb more of the material. If you were to take a average-sized human and extract their digestive tract, it would be around about 4 to 6 meters long, give or take. Now, if you were to take an organism that's roughly the same size, which is a predator, and take out its digestive tract, it would be proportionally smaller. Looking at a fox, it only contains one stomach. It has a short intestine, colon, and a small cecum here, before it leads out to the anus. The reason why is because it needs less time to digest our proteins. Now over here we have how an enzyme works. So the enzyme is our grayish area. The substrate is the material the enzyme breaks down. So it fits into it, into the active site here, like a lock and key model. But there is a little bit of space difference. Now over time the enzyme is going to break down the substrate into its individual products and then release them separately. Now if you did not have any enzymes in your body and you continued to have regular meals, you would actually starve to death because your body cannot break down that material fast enough. So even though you continue to eat regularly, your cells can't make use of it. Now, here we have the human digestive tract: liver, stomach, gallbladder, duodenum, pancreas, small intestine, large intestine, rectum, and anal canal. Now, there are different enzymes that are released throughout the entire body and are used for different purposes, each one highly specialized. And each of these organs are highly specialized as well, so ensure that you understand what each one is, what it does, and where it's located. The information over here will be very helpful. So pause the video and get it down. Alright. Nutrient and gas requirements. Now, every organism, whether it's an autotroph, heterotroph, herbivore, carnivore, detritivore, has requirements in order to keep the cells alive. If the cells do not have those requirements met, they begin to die off quite easily. So, both autotrophs and heterotrophs, they require gases and nutrients to maintain efficient and effective metabolic function. If they don't have air, they don't have food, they die. Inorganic and organic substances, water and oxygen gas. Autotrophs also require carbon dioxide. Heterotrophs need to take in all their nutrients. Autotrophs produce their own using energy from the sun, but they do need H2O, mineral ions, carbon dioxide, and oxygen. Autotrophs make their own glucose and other organic substances from inorganic nutrients. Heterotrophs must contain all of their organic nutrients by consuming other organisms. So obviously, big difference there. Heterotroph has to eat something else. Autotroph makes their own. Okay. Now, it's here we start to look at how materials are transported around the body. An open circulatory system is where the heart pushes and contracts in order to move hemolymph, which is a blood-like substance, not exactly blood, through vessels in order to bathe the organs. So imagine it like an open pool, okay? The heart is the water pump that is making sure that water is going into the pool and also leaving the pool. Now, the organs might be people that are sitting in the pool, just chilling out. Hemolymph is the water. So as the water / hemolymph is pumped into the pool, people relax, they go, this is really nice, and they're happy. But as the hemolymph / water leaves it, it goes back to the pump and then is sent back out again, constantly circulating. Hemolymph only transports nutrients and waste. It doesn't transport dissolved gases like carbon dioxide and water because those are transported through the sphericals. Okay? Insects are small enough they can get away with this stuff. Closed circulatory systems are used by much larger organisms like humans or dogs, cats, whales, bears, whatever. Because of this, the heart transports blood through a closed system. So it'll be much like how you can transport water throughout the pipes of a house. There's not necessarily going to be water exposed to the open air at any point unless you turn on a tap. But the water is still circulating throughout the house. Okay? The pathway of the blood and blood vessels goes around the body and back to the heart. So if you're starting at the heart, it goes to the arteries first, then arterioles, capillaries, venules, veins, and then back to the heart. Now in mammals, we have two types of transport systems, cardiovascular, which we just covered, and lymphatic. The lymphatic transports excess fluids throughout the body and goes back into the cardiovascular system. So for instance, if you're sick, your lymph nodes, like around your throat or under your armpits or in between your legs, they tend to get very enlarged because the lymphatic system also transports broken down diseases that the white blood cells have destroyed. The lymph nodes serve as like a collection point, and they try to get rid of it by flushing it throughout the body. The cardiovascular system is composed of red blood cells, white blood cells, platelets, and plasma. Each one of those have a distinct shape and role, which you can just see here. Please ensure that you've copied it down. Now, there are three types of transport vessels that we use throughout the body, or throughout a body like ours, in a closed system. They are arteries, capillaries, and veins. The descriptions for which can be found here. Please ensure that you pause the video and get that down, but also ensure that you find an image like this on the right hand side that compares fish, amphibians, what looks like a chicken and a human, and ensure that you get those down too. Copy and paste that from somewhere off your Google Images search, okay? Because we can see here that each one has a different style of heart. So a fish heart only has 3 chambers. An amphibian has 1, 2, 3, so right atrium, left atrium, and a ventricle. So it's also 3-chambered. 1, 2, 3, 4, 5. This is a 5-chamber heart, and over here we have the typical 4-chamber heart found in humans. Now, the human heart, like many other hearts, only serves as a pump. It's not a sign of romance, as you've been led to believe in many forms of visual art or media. It's a glorified pump. All it does is move liquid around. Now, doing so, it's actually one of the strongest muscles in the body. In particular, your left ventricle, which is responsible for pumping blood from inside the heart to everywhere in your body. So if we're following the details here, we're entering from the right atrium. For instance, the inferior vena cava and the superior vena cava. It flows into the right ventricle. It can't flow backwards because of these little valves here, much like inside veins. Instead, goes up here to our pulmonary artery, which goes to the lungs. The pulmonary veins transport oxygenated blood back from the lungs, and we can see here that's highlighted as a bright red color, meaning it's heavily oxygenated. Goes into the left atrium, which goes in the left ventricle, massive push out of the aorta. 30% of that blood automatically goes directly to the brain. The other 70% is distributed throughout the body. Alright. So this information already talks about the internal and external gas change. Please ensure that you pause the video and have a read through it. Okay. And we have a comparison between internal and external blood transport again. These two images, along with these descriptions, might be very helpful. And we're reusing similar images again. This image down here was a reptile, and that was a mammal. So reptiles have a 5-chambered heart. Cool. And we're going over the same information again. And again. And here we can compare between a fish, a salamander, and a mammal in terms of their orientation. The gill arches connect very quickly with our blood vessels here, whereas inside a salamander and a mammal they more likely compare to, say, lung connections. Now, essentially over time, the composition of gas exchange inside of blood vessels will change. When they start out from the heart, they have a very high oxygen content. When they finish, that oxygen content has been depreciated, whereas CO2 that started off as low has increased. That's not to say that it changes inside the blood vessel, but it's like getting off and on a bus. At the start of a bus driver shift, they might have a large amount of one particular denomination of people, maybe school students. During the middle of the day, it's mainly adults, and towards the end of the day, it's students again. This is based on where it's traveling and who's getting off and on. So in this picture, we can see the bright red representing highly oxygenated. Purple is a transition. Blue is low oxygen. Alright. It was a pretty intensive and sort of repetitive video, I know, but this information about how a living organism is organized is crucial to helping understand animal physiology and anatomy. So please ensure that you have a crack at these questions at some point when you finish the topic. If at any point you don't understand something's being covered, talk to your biology teacher, talk to your biology tutor, talk to other biology students, or leave a comment and we can try and address it in a future video. As always, I've been Forry, you guys have been amazing, and I'll see you around.